Hi, this is Nafisa. We are Group 10. Our project is about a step towards growing crops during the drought. I have Christine, Vanessa, Angeline, and Justin with me. Um, for the introduction, due to the increased global temperature, the precipitation and evaporation rates are increasing. As a result, the dry areas are anticipated to get drier and face droughts more often. According to NASA, the global temperatures has gone up by 1.18 degrees Celsius since 1880 and is expected to rise even more due to the human-caused climate change. Scientists have also said that the temperatures would not be constant throughout the world and that have been noticed in various places around the world as well. Thus, this would have a huge effect on agriculture and food supply. So in our project, our goal is to make a genetically modified crop that would be able to survive such extreme drought weathers as well as normal temperatures. So for reaching that goal, we would be using TDP gene to genetically modify crops to make them survivable in the drought or desert areas.
Hi, I'm Christina, and I'll be talking about methods. So to achieve our goal, we would insert the Trehalose 6-phosphate synthase, or commonly known as TPS gene, into the PBIN19 plasmid. The PBIN19 plasmid is a commonly used TI plasmid, which contains 2 antibiotic resistance genes, a bacterial promoter, as well as a plant promoter, making it an ideal plasmid candidate. It is a shuttle vector, meaning that it can be used in 2 different host species. The 1350 base pair alpha glutamine B1 promoter was found to be an effective promoter in rice, so it will be added to the PBIN19 plasmid, immediately preceding the TPS gene. The TPS gene will be inserted into rice in the following steps. The Trehalose 6-phosphate synthase TPS gene will be extracted from the U tardigrade species, Hypsipyus dujardini. The TPS gene will be used as a template for PCR amplification, along with the appropriate TPS gene primers. After amplification of the TPS gene, we will insert it into the PBIN19 plasmid. The plasmids carrying the TPS gene will be introduced into Agrobacterium tumefaciensis, which will then undergo antibiotic selection. The PBIN19 plasmid contains kanamycin resistance genes, so antibiotic selection will be carried out by placing the Agrobacterium into an agar plate containing kanamycin. The Agrobacterium that have successfully integrated the TPS gene will colonize on the agar plate and be used for further experimentation. It should be noted that the introns associated with the TPS gene have been taken out as the gene is being transferred from animal to plant. The Agrobacterium from the surviving colonies will be purified and inserted into Orza sativa. Agrobacterium will be used to transfect Orza sativa, commonly known as Asian rice, with the TPS genes.
Hello, my name is Justin, and I'll be talking about the purified DNA. So first, we got the coding strand from the TFAD website, and then we got the DNA template strand. And we then transcribed it to mRNA and then translated it into the amino acid sequence. So the first step to purifying DNA is to disrupt the cells to release the nucleic acid in the lysate. The second step is then to clean up the unwanted material and the cellular debris in the lysate through filtration, centrifuge, or other types of methods. Third step is to isolate the DNA by binding it into the matrices, and the methods of binding and purification will depend on the lysis. The fourth step is to then wash the solutions using the buffers like alcohol to remove the unwanted proteins, salts, and other contaminants. Fifth and final step is to use solvent like DNA elution buffer to wash a sample, and the purified DNA sample will be released.
So my name is Vanessa, and as mentioned, our plasmid is a binary agrobacterium vector, PBIN19. This plasmid will contain an origin of replication, a kanamycin resistance gene, a neomycin resistance gene, as well as a transacting replication protein that binds to and activates the origin of replication, the TPS gene, an alpha-glutenin B1 promoter, which is the region of the DNA where the transcription of the TPS gene will be initiated, as well as NOS promoter and an NOS terminator. I will also be discussing the ethical and legal implications of our experiment. When introducing the trehalose-6 phosphate gene into the Oryza sativa through DNA extraction of a tardigrade, it is important to consider the animal welfare, such as the organism's well-being and the invasiveness of the procedure. We will be using tardigrades as the test subjects, so the animal welfare is maintained at an adequate standard. These organisms go through anhydrobiosis when in a desiccation state, which allows them to adapt to stressful conditions. And in this ametabolic state, they have the ability to withstand environments with extreme temperatures and pressures. Our organisms will be experiencing these harsh conditions as protocol requires them to be desiccated in a desiccated state through starvation. However, this will not be an inhumane experimental technique as they could survive if they are rehydrated. Furthermore, as we are genetically modifying crops, it is also vital to address concerns such as how this GMO crop will impact other organisms and the environment, as well as its impact on traditional farming practice. More legal implications such as getting this GMO approved will address these. Agencies like the U.S. Environmental Protection Agency, the Food and Drug Administration, and the Department of Agriculture will verify that this crop will be safe for both humans and the environment.
My name is Angeline, and I will be discussing how it solves the issues along with our results. Over 70% of water is used for agriculture. When using these large amounts of water, it can lead to droughts. By having plants tolerant of desiccation, we will help these crops grow where droughts are more frequent and experienced. The areas that experience high levels of humidity and temperature will ultimately benefit from this. Those areas can include Ventura County, Northwest Los Angeles, and eastern parts of San Bernardino. We are trying to solve the issue by preventing plants from dying of water deprivation in these harsh environments, making it easier and less stressful for the farmers and individuals to grow crops. With our results, the crops would survive in drought weathers, allowing the individuals and farmers to be able to use less water, along with knowing the plants will have the desiccation tolerance genes implemented in them.
So to conclude our presentation, if our experiment proves to be successful, then we can use these methods and techniques to transfect other water-intensive crops and plants with trehalose 6-phosphate synthase gene, and that would give us the option to grow different other plants and other crops, not just rice. Thank you.